Well, good morning, everyone. Here we are once again. It's time for 3301. Bit of a different scenery today. I'm broadcasting to you from on location and away from my normal desk. And as would be appropriate whenever you go on vacation, you end up having your computer break. And it's happening to a lot of people very strongly. But so my regular computer is not working, so I'm using a different computer. So the lecture is going to be a bit bare bones compared to what we normally get. I won't have all the bells and whistles with my streaming setup, but I think we'll still be able to get through the lecture without too much problems. We love it, folks, don't we? We love having our computer break when we're away from home and can't fix it, and all our content is on there. Anyhow, it's a pain that I'm sure we've all dealt with at some level. So let's get started on our lecture for today, which is all about functional genomics. So this is, yet again, a bit of a review topic, although I think probably there's going to be some things here that you may not be familiar with. The kind of goal here is to discuss a little bit about how gene regulation works in kind of the context of modern genomics, just to, again, set the stage for understanding the different variants and mutations and so forth that we'll be learning about later on. So we'll get started. Now, before I really get into it, I wanted to tell you a bit about the conventions that we use in genomics and genetics more broadly. We talk a lot about genes in this class, and they're written down using a normal gene ID, but the species that you're looking at determines the exact way that you write it. So in humans, when you're talking about genes or transcripts that are from a human being, Homo sapien, the abbreviation is italicized capital letters. And proteins, the proteins that are derived from those genes or from those transcripts, are abbreviated in just plain non-italicized capital letters. So SHH in italics would refer to the gene that codes for Sonic Hedgehog, while SHH in all caps but not italics would be talking about the protein itself, Sonic Hedgehog. And now in mice, this is sort of important. Mice and rats and essentially non-human animals, or rodents at least, the genes and transcripts are actually abbreviated in italicized title case. And title case means that the first letter is capital, but the other letters aren't. And proteins are abbreviated as with humans in plain capital letters. So if you see SHH and it's written this way with capital S and two lowercase h's, you know that you're likely talking about the mouse version of that gene. While if you're seeing it in all caps, you're talking about the human version of that gene. So I think this is kind of important because in the assignments that I'm having you do, sometimes you'll be working with mouse data and sometimes you'll be working with human data, and you have to write things down appropriate for that species. And it's not really that I can't understand what you're talking about if I look at it. It's more just that this is the convention, and it's my job is to kind of enforce this on you so that you learn it and understand it as you go forward. So there's a few other naming conventions as well, but my own default, because I'm basically a mouse scientist, is to go back to the mouse convention and do the title case stuff. But we'll try to keep an eye on what we do to make sure that it's appropriate for every species. So why are we studying genomics? Well, I think you might, as I've shown in previous classes, there are some genetic correlates of mental illness that have patterns of inheritance that are somewhat tractable or compatible with the kind of Mendelian genetics that we've all been familiar with since high school. And the methods of classical genetics do have their limits. Candidate gene studies are, as I've said, not going to give you everything you need to know. If you have a specific gene in mind, you may be able to link it with a condition or a phenotype that you're curious about, but you'll never discover anything new by doing that. You can only kind of work with things that have already been described. And typically, the range of things that we can imagine and make hypotheses about is fairly circumscribed. It's kind of tied down with what, the things we already know about the brain, what neurotransmitters we think are involved, and what receptors and so forth. So there's some real limits there. But now, with genomics technology, we can study things on a genome-wide level with many thousands of subjects and look at every variant in the genome and ask, for each of these, how much do they relate to the phenotype of interest? So This, I'm sure, will be no surprise to you, but DNA is quite central to this. I don't want to, spend a whole lot of time about this, but just, to make the point, yes, the DNA exists and it's packaged into chromosomes that live in the cell's nucleus. Humans have 23 pairs of chromosomes and receive one pair from each parent. Different animals will have different numbers of chromosomes. Mice have 19 autosomes, and then, of course, the two sex chromosomes. Humans have a different number. Sometimes you'll get, weird animals that have, hundreds of different chromosomes. Others of them just have, a few giant chromosomes. The way that the DNA is structured into chromosomes, in some sense, is, biologically arbitrary, because whatever number of chromosomes you have, your biology will make it work and figure out how to get the genes it needs expressed. So, but this is the situation with humans, and that is kind of relevant for our world, because we want to know what chromosome a gene is on. So DNA is, sort of like protein in a way. It's a polymer. It's a chain of simple molecules. The 4 nucleotide bases make up DNA are adenine, thymine, guanine, and cytosine, ATCG. One of the kind of properties of the way science is done in the genomics age, and the way that, I tend to think of things and talk about things, is that the chemistry of it kind of takes a backseat to just the data side of it. So you don't need to really think about purines and pyrimidines and adenine and thymine and all that kind of stuff. Really, ATCG or ATGC, that alone will tell you almost everything you need to know about how the genome is structured, because ultimately it's like a data. It's a way of formatting data. It's a way of storing information, and so you would look at it in some sense the same way you would look at a text file or some code you've written on the computer. You don't even need to get into the molecular biology of it too much. And genes are segments of DNA that code for particular proteins. Although that even is a little bit controversial, because you would, in common parlance, a gene could also code for a long non-coding RNA. Non-coding RNA, of course, doesn't make proteins, but it does do important things in the cell. It could code for a microRNA, although, typically you wouldn't necessarily call those genes, but that kind of distinction is still fairly murky, because people say that they're studying genomics, and that includes everything in the genome, not just the genes. So it's a little bit murky there. So humans have somewhere around 20 to 25,000 protein coding genes. This is the number that's currently accepted today. It's probably pretty close to accurate, but it is funny to look at the history of genetics and the different guesses that had been made prior to the sequencing of the entire human genome. People used to think that the humans might have 100,000 genes or 1 million genes, or the numbers. Typically, when they were guessing, people would guess much higher numbers than what we actually appear to have, because human beings are so complicated. You would think, well, there must be a lot of genes in there that can achieve such a complex organism, and you would certainly think that humans must have more genes than, say, a mouse or some other sort of more basic kind of mammal. But that doesn't seem to be the case. Humans have about the same number of genes as most other mammals. The difference really is in how they're regulated. And certain types of genes have become more complicated and code for more complicated proteins. But for the most part, the big difference is just how the genes are regulated. The genes themselves can be a few hundred base pairs in length to several megabases, or millions of base pairs. And if you were to just download the human genome as a text file, like a plain text file on your computer, it would be about 2.9 gigabytes, which is actually pretty close to the size of the mouse genome. So, on the scale of pure size, the human and the mouse genome are not terribly different, which, you got to kind of wonder about, because human beings, naturally, appear to be much more complicated and interesting than mice are, yet their genomes are fairly similar. So let's look at the role of mRNA. So, of course, DNA can't be made directly into proteins. You need to transcribe it into messenger RNA first. And this is the first step of gene transcription. So during this, a given gene is copied into a strand of messenger RNA, the sequence of which is complementary to the sequence that it was copied from on the DNA. The difference, of course, being that RNA, instead of having thymine, it has uracil. It's a one-to-one swap that doesn't affect the meaning of the sequence, but it is somewhat relevant just on the level of the biochemistry of it. And then the next step, of course, is when messenger RNA is translated into proteins by ribosomes. If it happens to be a coding RNA. If it's a non-coding RNA, then it does other stuff. So, just to bring you back to, I've got this thing set to follow its own timings. That's why it keeps advancing. Anyway, so we all know the story about gene expression, right? So the DNA molecule is partly unraveled. A strand of messenger RNA is transcribed from it. The mRNA meets up with a ribosome. The ribosome uses transfer RNA to bring the appropriate amino acid in. The amino acids are determined by codons in the sequence. A codon is a letter three nucleotides long, and it just specifies which amino acid belongs in which position, and that's added to the growing amino acid chain, and ultimately a completed protein is ejected and then folded up and subjected to various kinds of post-translational modifications. So that's really all there is to gene expression. I'm sure that's nothing new to you folks. What I think maybe needs to be emphasized, though, is not how that works. That's fairly elementary, and in fact, it almost doesn't even, to be honest, like, you don't really need to know a huge amount about how that works on a biochemical level to get through this class. But it is important to really think about how this is controlled. That is perhaps a deeper question that's more relevant to our material, because, like I said, humans and mice and chimpanzees and all kinds of other animals have about the same number of genes, and our genomes are about the same size, yet we reach such different outcomes. Our brains are so much more complicated than any other animal on Earth. How is it that with the same basic set of genes, I mean, there are differences, but the same basic set of genes, how do we achieve such a completely different outcome? The answer is how the genes are controlled. It's not so much about what genes you have, but it's about what genes, or how the genes are controlled. And you can look at this on a number of levels, but one simple thing to consider is the fact that different tissues in the body express different sets of genes. So DNA is the same in every cell, but cells in your liver express liver enzymes, like enzymes for detoxifying things and for metabolizing nutrients that come in through the gut and so forth. And brains express different things. Neurons express different genes even from astrocytes. So every cell type within every tissue is expressing different genes despite having the same exact DNA. And what's more important, or they're also expressing different things at different times. A neuron is expressing different genes when it's developing than it does when it's mature. A neuron expresses different genes when it's dying than when it's healthy. There's a huge amount of variability in the timing and magnitude of all these things. So really the action, if you want to understand what makes us different at the level of species and also at the level of individuals, probably more of the action is happening just at the level of control of gene expression, not at the level of like the gene coding sequences themselves. So if you want to think about how gene expression is controlled, there's a couple of different parameters. And like I said, this is not going to be anything new or too advanced or surprising for you. This is maybe just a window into kind of how I conceive of things. There are basically three factors. You have location, where are the genes expressed? What cell types, what tissues? You have timing, when are they expressed? Are they sensitive to stimuli? Are they only expressed at a certain time point during development? That kind of thing. And then also quantity. How much of that mRNA and that protein need to be produced? Some genes are always there, but they're expressed at very low levels. They're just kind of floating around in the background. They might make a transcription factor or some other kind of thing that you don't need a whole lot of to have an effect. Others, you need vast quantities of this thing being produced all the time. This would be things like collagen or structural proteins that hold your body together. You need very large amounts of that stuff. And so the mRNA for that is always being cranked out in high quantities. So you got location, timing, and quantity. These are kind of the three main factors when you're looking at gene expression. And notice, again, we're not talking about the coding sequence for the protein. We're talking just about where it keeps advancing, sorry. We're talking about where and how much it's expressed in. So different parts of the brain are going to express different genes. This is part of the story when it comes to where. So, and of course, we're in the brain where, if we were studying the liver, all you'd really have to worry about is the liver as a whole. But because the brain is so much more complicated, it's not the brain as a whole. Every part of the brain is functionally and structurally different, and thus is expressing different sets of genes all the time. Oh, so Alexis St. Pierre asks, and sorry I can't bring up the chat window. Like I say, I'm running a very bare bones streaming operation this morning. I'm on I'm on a laptop that I borrowed, so I don't have all my, fancy technology stuff working properly. What would be an example of an mRNA or a protein that you don't need all the time? I know you said collagen is one that you would need all the time. It depends on what tissue you're in. I mean, you don't need it in your brain, but you would need it in like your cartilage and muscles and so forth. I know you said collagen is the one you would need all the time. I just don't know what the opposite example would be. Well, an opposite example of a gene that you don't need all the time would be a gene, for example, involved in apoptosis. So there are like BSX, for example, or BCL1. These are genes that are involved in basically causing cells to self-destruct. So, apoptosis is kind of this controlled cell suicide where cells carefully, kind of break themselves apart. It's like you're moving and you're trying to take apart your IKEA dresser. And you just take it apart piece by piece and package everything away and do kind of a controlled demolition. Those genes are only needed if that's what the cell wants to do. If the cell is not interested in committing suicide and blowing itself up, then those genes aren't expressed because you're in a steady state. So there's lots of scenarios where there's genes that, yeah, exactly. So there's lots of scenarios where there's genes that might be good at one moment but aren't good at another moment. Developmental genes would be the same way, right? There's genes that are involved in promoting, for example, neurogenesis and cell division. Actually, maybe this is an even better one. So there's genes involved in cell division that you need during development to have all of your cells divide. But it's very important that those genes are turned off when you're fully grown and mature because your cells are supposed to stop dividing. If your cells suddenly turn those genes on and begin dividing again, that would be an example of cancer or a tumor. Those genes would be called, like tumor oncogenes or proto-oncogenes. So like genes that are turned on, if they're turned on when they're not supposed to be, give you a tumor. Those are oncogenes. And so there's tons of examples like this. You could kind of go on down the line. You really want to have gene expression under a nice tight control. So different parts of the brain have different proteins, and that's part of, and different genes being expressed. And that's part of the reason, or that's part of why it's possible for different parts of the brain to have different functions. Places in the brain that synthesize dopamine, for example, are going to express tyrosine hydroxylase, TH. Parts of the brain that don't are not going to express it. And you can kind of go down the line, and there's tons of genes that are very localized to specific parts. Of course, there's plenty of genes that are universal to all parts, but typically there's kind of a correlation between the anatomy of the brain and gene expression. You can actually use the Allen Brain Atlas if you like to go and explore all of these connections. So this is one kind of level of gene expression control, right? You've got to figure out where the gene, like what type of cell this gene is even needed in, and then make sure that it's expressed there. There's also timings. As I was saying, like a lot of time, you don't need certain proteins. You only need them when you need them. And so gene expression is controlled in such a way that genes are only expressed when their proteins are needed. Now, this is probably the broadest category of all of these things because there's all kinds of stimuli that can control gene expression. You have things like neural or hormonal signaling. You have your physiological conditions. Some genes are only turned on when, for example, you are, hungry or thirsty, that kind of thing, to change your physiology around. Sensory stimuli coming in through your nervous system. Developmental programs, like I say, sometimes one gene can actually turn on another gene. There can be kind of a cascade of gene expression regulation. So there's a lot that goes into this timing category. I don't want to give you every piece of it now, but I just want to kind of make you aware that this is one of the things that's out there. And then finally, quantity. So it's possible for different stimuli to affect gene expression in a quantitative way. Genes can be up or down regulated depending on what it is that they do. So upregulation means that there's more mRNA being produced, and then typically you would assume more of the protein, although it's much easier to measure mRNA than it is to measure protein. So oftentimes researchers will just do the RNA analysis using RNA-seq or something like that, and then they'll assume that if there's more of a certain RNA, there must be more of that protein as well. I mean, having more RNA on its own doesn't really do anything. RNA is not, if you're talking about, RNA for an enzyme, RNA itself is not the enzyme. You can't exactly say with certainty that just because there's more mRNA that there's more of the chemical reaction that enzyme catalyzes. Or if you have, for example, mRNA for like a receptor, say for glutamate, just because you see more mRNA doesn't mean that receptor, there's more of it actually in the cell and that the cell is therefore more sensitive to glutamate. That's several steps removed from what you're measuring. But That's typically the inference that's made. The more mRNA, the more protein. And downregulation is just the opposite. If you have less mRNA, typically you would assume there's less of that protein. And therefore, whatever that protein does is happening less in those cells. So there's many examples of this, but I thought this was an elegant example. This is expression of the gene FOS. This is an immediate early gene. It's activated in response to stimulation of neurons. And here it is at baseline. You can barely even see it. This is in situ hybridization. So the darker the color means the more of this FOS gene is being expressed in the hippocampus and dentate gyrus. So at baseline, you barely see anything. And then after you expose the rat to a stimulus, you see a little bit more. And then as time goes on, you see even more. And this is an example of gene expression upregulation. So as time goes on, the level of this mRNA is increased, and theoretically you would assume more of the protein is also being made. So this is upregulation. And if you were to play this back in reverse, you would see downregulation. So that's all being said. Hopefully that's not going to be too new or surprising to anybody here. But what I kind of want to zoom out and give you a sense of is just how much regulation is being imposed on the genome and on the products of the genome. So we're here studying genetics at the level of variants and mutations that affect either the coding sequence of the gene or affect how it's regulated, whether it's through epigenetics or something else. There's many, many layers on top of that. So this is an example from this Bucatelli and Selbach paper from Nature Reviews, I guess. And so here are the different things that can influence phenotype. Phenotype is variation between people. So at the very, very basic level, you have genetic variation and genome architecture, how the genes are kind of put together, what order they're in, do you have any insertions or deletions, that kind of thing. Then you have histone modification. That's a type of epigenetics, as is DNA methylation. We'll learn about that in the next class. You have chromosome topology, like how the three-dimensional structure of the chromosome is set up. We'll also learn about that. And then you've got that. So that's all stuff happening on the level of DNA. Then you start to phase into RNA, where you've got transcription, and whether or not a gene is transcribed, of course, has something to do with its effects. And then you've got splicing. You can combine exons in different ways to create different products by alternate splicing. You have to bring the mRNA to where it's needed. That's mRNA transport. The mRNA can be modified by polyadenylation and other kinds of modifications. There's actually RNA methylation and acetylation, believe it or not. There's microRNAs. They can regulate whether mRNAs are expressed or not. There's mRNA degradation. And then you start to get into the proteins that are translated from the mRNAs. Then proteins can be folded in different ways. Proteins can be localized in different places. Some proteins go into the nucleus, some go to the cytoplasm, some go to the cell's membrane. They can be phosphorylated or ubiquitinated, and finally they can also be degraded. So these are all the different layers that can control the ultimate end product of your genetics, right? Ultimately you're working toward having proteins, but there's so many layers in between the genes and the ultimate outcome. And then you stack on top of that the interactome, which is how all the proteins interact with each other. Typically proteins don't work alone, they work with other ones. The metabolome, you're thinking like all the different metabolites and chemical byproducts of our ongoing biochemistry. Probably you could put the microbiome in there too. Many, many, many layers on top of all of this, and all of this on top of everything else, and the genetics is what leads to phenotype. So I'm giving you this to give you a sense of just how complicated the overall problem is. We're going to be down here in this blue area. So we're going to be pretty much just talking about genome architecture, variation, chromosomes, histones, and DNA methylation. And that's going to be basically all, maybe a bit of splicing too. But that's about it. So I think that's plenty for one class, but I just want you to be aware that this is just where things start. There are many, many layers that sort of fold on top of this and add more complexity to the issue. So you kind of have to bear this in mind. You're only learn so much. You're only going to learn so much from just genetics and genomics on its own, because even if people are genetically identical or very similar, there's all kinds of things downstream of that can also vary. So Ali asks, so upregulation is like a positive feedback loop. I guess I don't know if I would really want to say that, because a positive feedback loop implies that things are out of control, meaning that the more of something you have, the more of it gets produced. And that can happen. There are feedback loops in the regulation of gene expression, but typically it's much more controlled than that. You make a certain amount, and then you stop, and then it goes down. And there's not necessarily this kind of vicious cycle that you would associate with positive feedback loops. If anything, gene expression is closer to being under negative feedback, meaning that if you have too much of something, the amount goes down, and if you have too little of something, the amount sort of is upregulated. So it keeps things are maintained at the level that's appropriate by negative feedback, not positive feedback. Although I think in a disease state or in a pathological state, positive feedback could be involved, but I can't think of an example of that off the top of my head. Okay, so now let's talk a little bit about the structure of genes. This is kind of a necessary baseline or starting point for this class, because we're kind of looking at genes almost like in an informational context, not so much as bits of biochemistry, but as objects that we can study in a kind of a rational way. So the typical gene is organized from the is laid out from the 5' to the 3' end of the DNA. This is kind of a genome browser sort of view. So if you've done the genome browser assignment, you might recognize this basic kind of idea that like you can lay out the genome in a long line, and then along that line you can put in different genes where they're located relative to each other. You can indicate the transcription start site with this kind of right-angled arrow, and the coding region of the genome, or of the gene rather, is given by a certain color. So the coding region goes all the way from the transcription start site to the transcription end site. And these are transcribed 5' to 3'. So the coding region is anything that, essentially anything in the gene body that contributes to the actual protein and is like a protein coding region. Now that's the simple view. That's the basic, those are the basic borders of a gene. And if you want to get a bit more complicated, it's like if you're looking at Canada and you want to know what the different provinces and capital cities of each are, this is what you would look at. So genes are made up of different parts. There is the 5' untranslated region and the 3' untranslated region. These are places around the TSS and TES that are transcribed into mRNA, but they are ignored by the ribosomes and not actually converted into a protein. Within the genes, you have exons and introns. Exons are the part of the gene that actually code for the protein, and introns are, actually the majority of genes are introns, or like the majority of the DNA inside of a gene is intron. Exons make up a fairly small minority of the actual sequence of a gene. But introns don't participate in protein coding. They are there for other reasons, but they mainly just take up space in a certain way. They're cut out of the mRNA, and they're not actually translated into proteins. And then so outside of the gene, you can look upstream and downstream. And downstream would be toward the 3' end, and upstream is toward the 5' end. Now, the upstream area tends to be more interesting to people because this is where a lot of the regulatory elements that actually control whether a gene is expressed or not live. So upstream of a gene, you have things like enhancers and silencers. Transcription factor binding sites can exist inside of those. The promoter region is a region that's needed. It doesn't actually code for a protein, but it's needed to actually help the gene be expressed. It's almost like it's like a flag in the genome that tells the enzymes and the transcription factors, here's where a gene starts. So there's a lot of other stuff going on in this kind of map of the in a typical gene map, and you can actually add more and more kind of ornaments and decorations onto this plot, but this is the basic structure, right? And so when you're talking about areas of the genome that regulate expression, you're typically looking toward the 5' end upstream of the gene, either in the transcription start site area, the promoter area, or even a bit further upstream. This is only generally speaking, of course. You can have trans regulatory elements that are quite a long ways away from the gene itself and still control how much it's expressed. So genes are transcribed as one continuous unit from the TSS to the TES, the transcription start site, to the transcription end site. But that long bit of RNA is what we would call immature RNA. It's been transcribed, but it's not quite ready to make a protein yet. What has to happen is the exons, which are, like I said, a fairly small actual proportion of the overall mRNA, typically, are spliced out and stuck back together. So the exons themselves are the only part that actually code for proteins. And it's worth bearing in mind that coding genes of the overall genome, coding genes are a fairly small fraction. Most of the genome is something else, and people used to call the rest of the genome junk DNA. That's not true. It's outdated. We'll learn about what's going on in the other parts of the genome, but most of the genome is not part of protein coding genes. And then within protein coding genes, the exons themselves that actually code for the protein make up typically a very small fraction relative to the introns. So overall, the actual protein coding component of the genome that actually makes proteins that, we're made of proteins. My skin, your skin, your hair, your neural cells, they're all made of proteins in large part. The part of the genome that actually makes that is only about 1% of the overall length of the genome. So if you think of like 2.9 gigabytes worth of data, only 1% of that is really going to code for proteins. The rest of it does other stuff. And so this does lead you to look at exome sequencing. So I think the last class I was talking about DNA sequencing and how it can be expensive and complicated to have so much data coming in. One of the things that people will do to try to work around this is what's called exome sequencing. This is a way of purifying out just the exons from DNA and sequencing just people's exons. There are ways, and I guess I won't get into this, but there are ways to take the DNA that you sample from somebody and extract just key parts of it, so that you're only looking at certain parts of the DNA, not the whole genome. Whole genome sequencing was what we were looking at, but you don't have to do whole genome sequencing. You can actually just look at the exons. And if you do that, it's much cheaper because you're talking about 1% of the genome relative, compared to 100%. And it's also more targeted because mutations to the protein coding regions are going to have a much bigger effect on phenotype than mutations to introns. And mutations to introns do a lot, and we'll learn about that stuff. But if you have a problem with a protein coding sequence, that's very likely to cause a noticeable phenotype that really makes a difference in your life. So exome sequencing is one kind of response to this basic fact that exons make up a tiny proportion of the genome, but they're very, very important because they actually code for proteins. So we've talked a little bit about the types of genetic variation that exist. There's different varieties, and each variety really could be its own course if you wanted. But single nucleotide polymorphisms are the type that we're going to be looking at the most often. This is where the identity of a single nucleotide is changed. It's the most common sort of genetic variation. There's about one SNP for every 1,000 base pairs, or for every kilobase of DNA. Non-coding DNA, I should say, so introns and intergenic regions. And there's about one for every 2,000 base pairs of coding DNA. So inside of exons, SNPs are about half as common, or that is to say, they're twice as rare. This kind of makes sense, actually, because if you think about evolution, most mutations are deleterious. Typically, you don't want to have mutations because they almost never help. However, once in a while, they're neutral or helpful. Now, the odds of a mutation having any effect at all are so much higher if they're in a protein coding region, because they could potentially affect the actual protein sequence. And it's not common that effect is a good one. So mutations are underrepresented, or variants I should say, are underrepresented in coding DNA because they're selected against, because the odds of them changing the protein and causing problems are much higher. And so natural selection kind of weeds those out. They're more common in non-coding DNA because the effects tend to be more subtle and they're not really subject to natural selection in the same way. Most SNPs have no real apparent effect on phenotype, but some are strongly linked to disease. Now, when we're talking about no apparent effect, it means no apparent effect at the level that we can detect. When you kind of zoom out and look at this on a bigger scale, even SNPs that on individual levels don't seem to have an effect do seem to synergize or combine with other SNPs and other types of variants to change your risk of disease. As Canna writes, so SNPs usually have no effect. Yeah, typically SNPs have no noticeable effect. They're just sort of harmless variants that don't really do anything. They make people very different. I mean, if you look at the differences between, say, population groups on the planet, people, say, who come from East Asia are going to be genetically very different than people who come from, say, Western Africa. But most of those differences are kind of meaningless from a functional perspective. They don't make them any different in a meaningful way. They just add variation because you've been separate people have been separate populations for a long time. Different kinds of variants become more common in isolated groups. But because most SNPs don't really do a whole lot on their own, most of that variation just leads to what we call population stratification. It makes people very different genetically, but it doesn't have any meaningful effect, or doesn't typically have an effect on much of their phenotype. Although you have to go in and look at this on a SNP-by-SNP basis. You can't say that no SNP does anything. You have to look. But typically SNPs are not having a huge effect on their own. They're just kind of variation. However, our goal in this course is to look at the SNPs that do make a difference, and there are some that do seem to predispose you to different kinds of diseases and conditions. So the default assumption is that they don't do much, but we find often that they do certain things. So Natalie asks, is there a reason why SNPs are the most common type of genetic variation? Is this reflective of the efficacy of the genetic machinery? Yes. So the DNA is copied very nicely with a very high degree of fidelity. And typically, either because of DNA repair or because of the fact that a larger mutation would be lethal, either to the embryo or to the person, larger scale mutations do not tend to be passed on. They don't tend to accumulate in the gene pool because they're usually harmful enough that they would take that lineage and just put it to an end. So SNPs are the most common because it's very, it's comparatively simple to have a single nucleotide get swapped over. It doesn't take, it's a fairly small kind of mutation, so it would happen at a higher rate. And the odds of it ruining the organism or causing real problems for survival and reproduction are lower. Therefore, they're more common because they have less of an effect and they're less selected against by natural selection. And then Lauren asks, are SNPs de novo mutations, or are they passed down through generations? Good question. So SNPs can be de novo mutations. Obviously, at one point they were, right? Somebody had to get the SNP in the first place and then pass it on. But typically, if you're talking about your SNPs, like if you're getting genotyped and somebody's giving you a list of all the different alleles you have for all of the different SNPs that are known, almost all of these would be passed on. You will get, every person has some number of de novo mutations that are unique to them, but the vast majority of the genetic variation that makes you unique is just passed down from your parents, and that would be mostly SNPs. So we also have indels. These are insertions and deletions. We've talked about these. This is when a fragment of DNA between about 1 to 1,000 base pairs is physically inserted or removed from a genomic locus. This can happen quite often. These are often, much more serious or large-scale mutations or variants, but they do set people apart quite often, and oftentimes they pose a risk for disease as well. And sort of one kind of version of this, or one kind of offshoot of this, would be what's called copy number variations. So typically, we would assume that everyone's DNA matches the reference sequence. So you would have essentially one copy of every gene. You'd get one allele of it from your mom and one allele of it from your dad. So essentially, there'd be two copies of that gene floating around in your chromosomes. However, it is possible for there to be copy number variations that for certain genes or certain regions of the genome, you can have multiple copies of it. Now, this is not terribly common. You have millions of SNPs, but only a few copy number variations. But it can be fairly relevant because it affects what we call the dosage of the gene. Like I said, most people have two copies of every gene, unless it's on your X chromosome, and then it's either one or two, depending on whether you have one X chromosome or two. But typically, for the genes that are located on autosomes, you would have two copies, and that's kind of what your body is expecting. The amount of mRNA that's produced influences the amount of protein that's produced, and all of that is regulated with the assumption that you have two copies of that gene. If you have extra copies, they might make more of the mRNA because now there's twice as many or more transcription going on from multiple sites. So it might be the case that you would have more of the protein or more of the mRNA that's produced by that gene. Now, this may not matter, but it might actually make a fairly big difference because it could have an effect on phenotype. If you're coding, say, for an example, an enzyme, more of that enzyme means that whatever it does is happening at a higher rate. And this can actually be beneficial in some cases for population-level adaptations. So here's an example from one of my genetics textbooks, and it's looking at copies of the amylase gene. Amylase is an enzyme that breaks down starches, and it's needed to break down kind of carbohydrates and refined starches and so forth. And if you look at the number of copies people have of this gene, it seems to vary based on their kind of ancestral diet. So here's a map, and people who in their ancestry would have eaten a high starch diet, a lot of rice and that kind of stuff, have more copies of amylase than people who would have eaten a low starch diet. A low starch diet would be things like, you'd have more protein, you would eat more meat and that kind of stuff. So it just depends on what the staple diet is of your ancestry. But, so yeah, take for example, down here, like you can look at people in Japan have more copies of amylase because rice would have been a bigger part of their diet than people up here in Manchuria or Siberia. They would have had more meat in their diet. And so there is a difference in the number of copies here, making some people more able to digest starch than others. And of course, you can think about lots of other examples here. People's response to, or people's ability to consume lactose, for example, after they finish being babies. That's not a copy number variation actually, but it's that same kind of thing. Like there are some areas where like your genetics and your metabolism have to do with what your ancestors consumed. But in this case, it's a copy number variation. Canna writes, could this happen with alcohol tolerance and the enzyme alcohol dehydrogenase? Yes. So the ancestral patterns of alcohol consumption do seem to be the reason why some people of different ancestry have, or there's variation, I guess, in people's ability to tolerate alcohol and whether they have enough alcohol acetaldehyde dehydrogenase and alcohol dehydrogenase. So that's another type of variation. Although off the top of my head, I don't know what the difference is there, but this is one of those same kinds of things, right? Now, let's take a look at how gene expression is regulated on a little bit more of a little less abstract level. So what we're assuming is there's some kind of, the context that a cell exists in kind of determines how much of a given gene is needed. As I said, there are many stimuli and scenarios that regulate gene expression. You've got timing, you've got location, you've got stimulation like exposure to growth factors or outside stimulators, all kinds of reasons that would influence whether or not a cell needs to express a gene. So put that aside for now and just ask, okay, once that's sort of been decided, how is that gene expression actually regulated on the level of the kind of molecular biology? And typically what's going on is gene expression is regulated by transcription factors. And transcription factors are nothing more than proteins that float around the nucleus and have the ability to bind to specific places on the DNA. When they bind, they are able to recruit enzymes like RNA polymerase. And once RNA polymerase is brought to the party, it will come in and make RNA out of the gene that's in that area. So if you want to regulate gene expression, what you want to do is attract transcription factors and a certain type of transcription factor called a transcriptional activator. You want to attract them to the neighborhood of that gene, typically toward the 5' upstream region, either in the promoter or in somewhere, a nearby enhancer or something like that. Those transcription factors will form a complex with RNA polymerase and a number of other proteins that ultimately result in that gene being transcribed into RNA. A whole bunch of other changes happen at the same time that we will get into, but this is the fundamental kind of level. If you want it, step one is bring all of the appropriate proteins and enzymes to the party and put them in position upstream of the gene to start transcribing. You can also have scenarios where proteins bind to the DNA and instead of activating transcription, they actually repress it. So there are some cases where you can have transcriptional repressors that decrease gene expression. And what they would tend to do is cause the chromatin to be condensed, to push the gene deeper into chromatin where it can't be accessed, but also to kind of repel or prevent the binding of RNA polymerase and all of these this transcriptional complex. Transcription factors bind to these things called transcription factor binding sites, TFBS. We'll hear about that in a few slides from now, but the basic idea is this. The proteins are made to kind of target or bind to specific sequences in the DNA that match with a motif that the protein is sensitive to. So these transcription factors can't bind to just any old place. They have to bind to a place that's ready to accept them. And those binding sites tend to be upstream of genes, and this is what enables these things to bind and actually regulate the expression. Now, transcription factors tend to work in a combinatorial fashion. It's a little bit more complicated than what I showed you. But typically, there's a whole bunch of these that all bind and work together ultimately to bring RNA polymerase to the promoter region and cause it to start transcribing. So This gets fairly complicated because if you really want to understand, therefore, how it is that a gene is being expressed, you have to know about, well, what transcription factor binding sites are near that gene and which of those happen to be bound with transcription factors. The combination of transcription factors all results in sort of a combinatorial effect that changes the actual kinetics and likelihood that a gene is expressed. You can have repressors that are in the mix as well that can slow things down. And of course, epigenetics factors in here as well, which we will get into. But this is the basic logic. To express a gene, you need to bring all of these factors and enzymes to the party and have them sitting there at the promoter region of the gene, ready to go to town and start copying or start transcribing that gene. And this can involve, like I said, binding sites and regulatory sequences inside the promoter region, which is sort of maybe about 3,000 base pairs flanking the transcription start site, so fairly close to where the gene actually starts. And there can also be distal regions called enhancers that are located a good distance away. Proteins can bind to this and fold the DNA up in a way that creates this rather complicated three-dimensional structure that, as always, regulates expression. So transcription factor binding is sequence specific. So I wanted to kind of make an analogy here to give you a sense of what I mean by motif. Because transcription factors bind to motifs. These are short sequences of DNA with a stereotyped but somewhat variable sequence. So these are present throughout the genome, but they're kind of enriched in regulatory areas that are upstream of genes. And what I mean by a motif is a sequence of nucleotide, a sequence of letters that is very similar and repeated all over the place wherever this protein happens to bind. But it can be similar, but not the exact same. So I kind of put this sheet music up here to give you like an example of what I mean. So a motif would be like in music, if the same pattern of melody appears multiple times through the score or through the music. So like I guess one example would be like in Star Wars, that kind of Darth Vader song, like dun, 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 like that thing. That little piece appears all over the place in the movies because whenever they need to kind of remind you that Darth Vader is a bad guy or like the Empire or whatever it is that song is supposed to be about, they'll play that all over the place, maybe with a different in a different key signature or with a different timing or maybe with a few variant notes here and there, maybe on a different scale potentially, but it's the same motif, and it's the point of it is that you're supposed to recognize it and make an association with a given character. I gave one here that from La Cucaracha, which is a kind of a traditional song. But like here, the first part of this, it's the melody is played over the E flat chord, and then it's played again over the B flat 7 chord. And every note here is different, but it sounds kind of the same. There's just a few differences because it's played over a different chord. And that's another example of a motif. It's very recognizable, but it's somewhat different. It's the same kind of riff, but it's a little bit different. So this is, now let's take that and apply it to the question of transcription factor binding sites. So these are, transcription factor binding sites are characterized by motifs that bind to particular transcription factors. And this is what I want to show you. This is kind of what I want to represent. And I think maybe it's a little bit abstract to think of this, but think about the way that DNA forms base pairs. There's a kind of a complementary nature there that allows DNA to hybridize to itself. Well, it's also possible for proteins to bind to DNA. Now, of course, they're not binding through base pairing. Proteins don't have base pairs like DNA does. They don't bind that way. But they do have the ability to recognize specific sequences. There are proteins that have sites on them that can recognize particular sequences of DNA. And wherever they see that, they will stick to that part of the DNA. So that would be a transcription factor binding site, and those binding sites are characterized by motifs that describe what the sequence there is. And we can represent that visually with what's called a sequence logo. So these colorful little rainbow things with the letters, that's called a sequence logo. And in a sequence logo, the height of each nucleotide corresponds to its relative importance for transcription factor binding. So, as I said, motifs can be somewhat variable. It doesn't have to be a letter for letter identical every place you look, but it has to have certain key patterns. So, an example would be here. This is a sequence logo for the Jun AP1 transcription factor complex. And the height of every letter determines how important it is. So letters that are given at full height have to be there. So here, you absolutely have to have a T, a G, and an A. You absolutely have to have a T, a C, and an A. But then in between them, you could have basically either a C or a G. You can see the C or G are about equal size. They make up about 50% the height there. That means in the middle, you could have one of the two. It kind of statistically doesn't matter. And you can discover these through a kind of a computational analysis of chip-seq experiments. This is where you're using sequencing to pull down parts of the DNA that are bound to specific transcription factors. And once you have the sequences of the DNA that you know are bound to those factors, you can ask what motifs exist on those places. And so the way you would arrive at a logo like this is you would see in your sequence, you'd see TGA, and then you'd see TCA, and then so that would appear everywhere. But then about 50% of the time you would see a G, and then 50% of the time you would see a C. So you basically would arrive at a sequence like this. TGA has to be there. CG, 50-50. It could either be a C or a G. They're present in equal amounts. And then TCA has to be there. That's how you read a sequence logo. So I think this is the kind of thing, like, I hope that what I'm telling you is making sense. The way this is trying to represent the concept of a motif. This would be the kind of thing that I probably would ask on an exam because it's an easy question. If I give you a sequence logo, I just ask you to tell me what it is and interpret it. So I'll give you a little hint there for an easy question that I might think of. Because these are quite relevant, right? I mean, I think they really make the point that transcription factor binding is sequence specific. And if you know where those sequences are located in the DNA, you have a sense of where those transcription factors can bind and therefore what genes they might be capable of regulating. And that all adds up to a fairly important picture. So I'm just going to take a break and get a glass of water. I'll be back in a few seconds here, folks. I'm just getting a bit of a dry throat. And apologies, I can't play my my fancy waiting time music because again, my computer's not working. Okay, and we're back. Sorry for that, folks. Sorry to keep you waiting. Anyone have any questions before I get started again? Or does this all make sense? Okay, so that's about all I wanted to say about transcription factor binding, and I think that hopefully the idea of the sequence logo makes sense to you. But again, just to emphasize, what it means is transcription factor, for every transcription factor, it's possible to learn what nucleotides have to be there in order for it to bind and what nucleotides are kind of optional. And the hint that I would give you is that these are places where you really don't want to have a mutation, because if you imagine one place where you were to change the G to something else, that transcription factor could no longer bind there, and that would have a pretty big effect on expression. Another thing that I wanted to tell you about is enhancers. So Speaking historically, it was thought that gene expression was mostly regulated by nearby elements. So you would look to the promoter region of the gene, the region kind of immediately flanking the start site of that gene, and assume that everything that was happening there was all that could regulate the gene. But now it's known that genes can be regulated by very distant elements called enhancers. Enhancers may be tens to hundreds of thousands of bases away. They are located so far away from a gene, it's very tough to even link an enhancer to a specific gene without empirical validation. But the way they essentially work is transcription factors bind or can bind to enhancer regions, and then the DNA will physically fold itself in such a way as to bring that enhancer, that transcription factor bound enhancer, toward the promoter of the gene in the transcription start site. And that three-dimensional interaction that occurs over a very long distance takes pieces of DNA that, like I say, are far apart spatially, or far apart in the sequence of the DNA, and brings them very close together spatially. And in this way, transcription factors can bind and regulate the expression of genes through this mechanism. So we'll get into that a fair bit more to explain this more, but it's a very interesting dimension of gene expression regulation, this long-distance interaction. The genome is spatially organized. So as I've been kind of hinting, there's a three-dimensional structure to the genome. Every chromosome is a single uninterrupted molecule of DNA going from position 0 all the way to maybe 250 million base pairs. If chromosome 1, which is the longest one, were extended to its full length, it would exceed the diameter of the nucleus by 15,000-fold, meaning that you can't just have the DNA in one long line the way it looks on the genome browser. It has to be kind of scrumpled up and folded away to fit inside. It's a little bit like the way your cortex works. We all learned that, of course, your cortex is wrinkly because it needs to fit into your skull. So, the cortex is like a sheet, right? So if you have a sheet that you want to make smaller, you kind of scrumple it up and give it all kinds of folds and wrinkles, and then it can fit into a smaller space while maintaining the same level of surface area. The DNA is the same way. You have to carefully package it away inside the nucleus and keep all of the bits of it organized. You can't just it can't be like the box of computer cables that you have that's all tangled up. It has to be more like rigging on a ship where it's nicely organized and folded up in multiple stages. This is part of what keeps chromosomes organized during metaphase, and also regulates gene expression during the interphase. And of course, neurons are basically always in interphase. So the three-dimensional spatial organization of the chromosomes and the genome as a whole is a very important topic because, as I mentioned with enhancers, sometimes the spatial organization plays an important role in gene expression. If you have a transcription factor that's bound all the way over here, but it comes into contact with the start site of a gene by three-dimensional folding, then that's a long-distance relationship that's mediated by this very delicate spatial organization of the genome. So DNA is wound around these things called histones. Histones are proteins that are put together as an octamer. There's 8 pieces to them. To create what are called nucleosomes, these little kind of beads on a string. And histones can be post-translationally modified. We'll learn about this when we talk about epigenetics. But this will change their relationship with the DNA and alter gene expression. Nucleosomes are wound into fibers and then wound again into bigger and bigger fibers and coils and so forth, ultimately resulting in the three-dimensional structure of the chromosomes. But all of this careful organization means that the actual genome is configured in a very precise spatial way. Where a gene is located in this 3D structure makes a huge difference as to how easily it will be expressed and where it will be expressed. If a gene is all packed away, buried underneath like several layers of other bits of DNA that have been folded on top of it, it's very difficult to express that gene. On the other hand, if a gene is like sticking out and has pieces of it flapping in the breeze, it's rather easy for that gene to be expressed. And so where a gene has been physically placed in the three-dimensional structure of the chromatin, or of the chromosomes, or the chromatin, makes all the difference for how well it can be expressed. So during interphase, the the three-dimensional structure of the genome is not in those kind of classic chromosomes that you think of as metaphase that you would have a karyotype of. It's more of like a continuous sort of fluid that's all the chromosomes are kind of bound up together in this large-scale structure. So there's a three-dimensional rendering of it here in a non-dividing cell, so a cell that's in interphase. All the different colors in this three-dimensional model represent separate chromosomes. So you can see how they're all kind of like mixed in together. They're in their own spatial domains, like they're not connected exactly, but they're all kind of mixed together into this like three-dimensional structure. We're lucky in a sense that neurons don't divide, so we don't have to worry about the different phases of mitosis. We really just have to worry about the steady state part that we call interphase. So, as I was saying, DNA accessibility is a very important part in regulating gene expression. If you have bits of DNA that are accessible, meaning that they're located toward the surface of this clump and enzymes can get in and reach them, then they tend to be easily expressed or expressed at high levels. On the other hand, if you take genes and put them deeper into the three-dimensional structure, where they're out of the way and enzymes and transcription factors can't get at them, they tend to be expressed at a lower level or not expressed at all. So actually, when you're looking at how you're putting together cells and organizing what cell types are what, one of the ways you can control, one of the ways you can make sure that neurons only express neuron genes is for neurons, all of the non-neuron genes have to be packed away so that they can't be reached by the enzymes. On the other hand, if you have a liver, all the liver enzymes would have to be accessible. So the DNA accessibility, how easily these enzymes can reach their target genes, makes a big difference for expression, and this is one of the main aspects of epigenetics that regulates the differences between cell types. All cells have the same exact DNA, but how the DNA is organized and regulated is what makes cells different from each other. Now, we can look at different cell types, for example, neurons versus astrocytes. They're both expressed in the brain, yet they have thousands of genes that are differentially expressed between the two. Now, how is that possible? They've got the same DNA. How could they be so different in terms of their gene expression? Now, one kind of naive view of this would be that every gene that's differentially expressed has its own transcription factor. That transcription factor regulates that one particular gene, and so you would just have to explain for, you've got thousands of genes that are differentially expressed, then you've got to differentially express thousands of transcription factors. You kind of create this like endless loop where all of the there's no kind of hierarchy. Every gene is regulated by one other gene, and then those genes, of course, will be regulated by one other gene, and this kind of this like endless recursion where there's no top-down control. There has to be a more general way to regulate gene expression differences between cells. Now, some transcription factors are extremely cell type specific and others are more generalized, but what really matters is the synergy between them. Transcription factors bind to their regulatory sequences in a combinatorial fashion. So you can have some that are very cell type specific binding alongside other ones that are very general. And it's the combination of transcription factors working together that really determines the outcome. So there are about 1,600 transcription factors in the entirety of the human genome. So 1,600 transcription factors, there's about 20,000 some odd genes. But when you think about how many combinations you can make out of 1,600, there's vast quantities of different combinations that you can have, meaning that you can regulate genes in a rather varied way, especially because it's not just combinations of 2, it's combinations of 3 or 4 or 5 or whatever number. So Alexis asks, sorry, can you go over those two bullet points, those 2 bullet point questions beneath the first point again? Right, so I guess what I'm trying to ask is, imagine you're trying to explain how it is that there are thousands of genes that are differentially expressed between neurons and glia. Glia, for example, express MBP, myelin basic protein, or not astrocytes, I guess, but like astrocytes express GFAP. And neurons express dopamine receptors or something. So there's all kinds of genes that are different between the two cell types. And like, how do you have thousands of differences? Do you need a separate mechanism for each difference that explains that difference only? Or is there a more general mechanism that can explain all of the differences all at once with just one thing? That's the question, right? Now, I'm asking this kind of rhetorically because we know the answer is that there's really just a few separate mechanisms. There's just a few mechanisms that enable all of those thousands of genes to be differentially expressed. It would be impossible, really, for there to be thousands of separate mechanisms, one for every gene, because it would just lead you to this endless recursion. If there was 1,000 mechanisms for all of the thousands of genes that are different between neurons and glia, each of those 1,000 mechanisms would need something else upstream of them, making them different. You see what I'm saying? You need to have a kind of a more hierarchical thing where there's one difference that feeds into multiple differences that kind of cascade down. So what I'm getting at is that cells can be different because they express different transcription factors, and they express different transcription factors in different combinations. So there's 20,000 genes. There might be thousands and thousands of them that are different between cell types. You can achieve all of those differences with just a handful of transcription factors, because transcription factors can regulate many, many genes at the same time. And so if you have one transcription factor that's only for neurons, all you have to do is have that one transcription factor be higher in neurons than in other cells, and it will find all of the genes it needs to and turn those on. So transcription factors are kind of grouped in a way that's like vaguely functional. There are transcription factors that are particular to every cell type. If you turn those on, they'll go out and look for the genes that they need to turn on to make that cell type what it is. And then when you combine other transcription factors into the mix, you can create these very complicated recipes. I mean, it's a bit like, well, I mean, it's, you know, it's kind of like how, you know, you can take flour, butter, eggs, and you know, baking powder and make it into all kinds of different cakes. You know, the same ingredients in different amounts can produce all kinds of different outcomes. So transcription factors are really the secret to this. You've got many, many genes, and orchestrating which ones are turned on and off at the same time is the job of the transcription factors. And because there's only a few transcription factors, you can get a lot of complexity just by combining them in different ways. So I'll give you kind of a practical example of this from the developing spinal cord. Now, this example, I hope this comes out clearly to you. But if you remember learning about neural development or about embryology in general, you would remember that gradients of growth factors make a big difference in determining what cells do. Cells in the developing nervous system, for example, are able to kind of sense the level of things like sonic hedgehog, SHH. They can kind of sense how much of it there is, and depending on how much they're exposed to, they will express different patterns of transcription factors. So, sonic hedgehog is secreted from the floor plate, I guess, from the ventral part of the developing neural tube. And so, anywhere toward the ventral part of the developing neural tube is going to be exposed to very high levels of sonic hedgehog. As you move further away from the floor plate, the level of sonic hedgehog goes down. So you can tell essentially where you are as a cell by looking at how much sonic hedgehog you're exposed to and determining and basing everything from that. And so, and I don't want you to fuss or worry too much about the anatomy or this exact example, but I'm trying to kind of give you a thing you can kind of generalize and understand as a kind of a rule of thumb or a general principle for how this thing works. Cells that are exposed to very high levels of sonic hedgehog are known to express NKX 2.2/2.9 and NKX 6.1 and 6.2, but they don't express OLIG2. On the other hand, they don't express PAX6 or IRX3 or DBX3. So every these by the way, I should say these are all transcription factors. Every transcription factor in this case has a certain amount that it's either induced or repressed by sonic hedgehog. So the amount of sonic hedgehog that's around determines what transcription factors are expressed. If there's a lot of it, you'll get a lot of NKX 2.2, but very little PAX6. And so every cell along this dorsoventral axis is going to be exposed to different amounts of this growth factor, sonic hedgehog, and will therefore express different combinations of transcription factors. So the example I've given here, P3 cells, the most ventral, they express high levels of NKX 2.2, 6.1, and 6.2, but then low levels of PAX6, IRX6, and DBX3 and PAX3. So don't worry about that exact part, but just know that they can basically convert this very basic form of spatial information, how much of this sonic hedgehog are they exposed to, they can convert that to a pattern of transcription factors that they express. And because the transcription factors bind to specific places in the genome and activate certain genes, all these cells can end up being different because they've got different genes being activated. This creates this kind of cascade where like you start out with a few differences and they kind of snowball into bigger and bigger differences. And this is what allows cell types to differentiate through transcription factors. Transcription factors are downstream of fairly simple signals. They add a lot more complexity because the combination can regulate all different combinations of genes, and those combinations of genes lead to differentiation of cell types. So you see, the development of the nervous system and the body in general is really a process whereby fairly simple signals and fairly simple sets of cells are made progressively more complicated by adding layers and layers of additional information and regulation on top of it. You start out with one cell, it turns into like a zillion cells. All of them are different. This is how it works. You add bits and pieces of information, and this combinatorial aspect enables so many combinations to be made out of a fairly small toolbox of transcription factors. So there's another point that is kind of similar to this but not super related, was Conrad Waddington's epigenetic landscape. So I don't want to confuse you by implying that this is talking about epigenetics like we'll talk about in the next class. This is from a different time when epigenetics meant a different thing, the old-fashioned meaning of it. But the basic idea is that cellular differentiation, in his mind, was a bit like a ball rolling down a hill. As it moves down the hill, you can drop a ball from the same place, and as it moves, it'll bump into different peaks and valleys and troughs and hills and whatnot that will eventually force it down particular paths. It'll make branches and go in one direction, and then it'll hit another branch and go a different direction. And so even though you could drop 1,000 balls from the same starting point, depending on where the branches are, the balls will end up in very different places. And this is his analogy for differentiation. Cells all start out the same, and as they differentiate, they become increasingly specialized. A cell begins as basically an ectodermal cell in the developing embryo, and is eventually converted into a very specific type of neuron, say a medium spiny neuron in the striatum. How does this happen? By successive branching of possibilities. So these branches, where a cell hits a certain point and has to decide whether it's going to go this way or that way and become increasingly differentiated, those are established by combinatorial patterns of transcription factors. So epigenetics and transcriptional regulation play a role in this process of development, where cells move from being very general all the way down to very specific. Every step of the way, there's different transcription factors that are activated, turning on different sets of genes that make this all happen. So you can kind of elaborate on this original hand-drawn thing by imagining that the hills and valleys themselves are established by different transcription factors. So if you go one way, you're exposed to the green and the blue and the red transcription factors, all of which can steer the cell in one direction. And if you go a different way, you're exposed to a whole different set. So the cell begins in a very general way, and by the combinatorial actions of transcription factors, gradually takes on a more and more differentiated state. So what's all this talk about? Why am I emphasizing transcription factors so much? Well, we're trying to understand how fairly subtle differences in genetics and genomics, genomics and epigenomics, or genetics and epigenetics, result in differences in phenotype. Now, I think I've already hinted that most of the genetic variation that exists in humans is in non-coding regions. So right off the bat, we have to kind of wipe out protein coding differences as a reason why people can be different, or a reason why a lot of diseases exist, because protein coding variants do not explain very much in the big picture. You're not going to find out what causes schizophrenia by looking for protein coding variants. Those, as far as we know, those don't exist, or they don't exist in any kind of meaningful levels. You got to look at the places that aren't protein coding, and the only way a place that doesn't code for a protein can have any effect at all is by regulating gene expression. Maybe it doesn't affect the sequence of the gene itself, but if it can change how much the gene is expressed, where it's expressed, what splice variants might come out, that kind of stuff, then that can explain how that gene links up with phenotype, or how that variant affects phenotype. Now, places where transcription factors bind are crucial, because if you have a mutation that occurs where a transcription factor binds in one of those motifs, then that can have a huge influence on the expression of that gene, much bigger than it would if it were a mutation that happened somewhere else where there was no binding of a transcription factor. So we want to understand where are the places in the genome where small variations have a huge effect, or have a much larger effect on the molecular biology of the cell by binding to places, or by existing in places where transcription factors and other things bind. So we're trying to link the molecular biology of gene expression with with variation in the DNA sequence, and then later on we'll also stack in epigenetics and other sorts of things. So we want to learn about how the genetic and epigenetic variants we study influence the molecular biology of the cell. And so much of that comes down to transcription factor binding and these networks of transcription factors that's really has to be emphasized, and before we can move on any further. So I guess the one last thing I'll give you is kind of an analogy. where I'll point out that regulatory elements in general, and regulatory elements is sort of a broader category encompassing enhancers, transcription factor, binding sites, and various other things in the genome that act to regulate the levels of gene expression. Mutations here do not affect the code or the protein coding sequence of the gene, but they have a large effect on how well it works. And so I was kind of thinking, like, a mutation here is kind of like if the handle breaks off of your door. It's a very small piece of damage overall. I mean, you know, it's not like your whole car is broken, but it's a key point. It's a point where you have to interface with the door or the car in order to be able to use it. So mutations to regulatory elements where there are where gene expression regulation is actually happening, those have a big effect on the cell despite not changing the protein coding sequence, because they're where all of the enzymes and transcription factors want to bind in order to make things happen. So this is a concept you have to be aware of, because it's just very relevant to understand all of the different variants and mutations we're going to be talking about as we go forward. All right, so that is it, folks. That's all I had to say for you today. Apologies for the kind of different format. Like I say, I'm not on my actual computer. I use this I do these streams with OBS, which is the kind of same sort of streaming software that like Twitch streamers use, I think. And it was all really dialed in very nicely on my laptop, but like I say, my laptop is broken and it's going to Apple today. So, you know, we'll see. But hopefully this will be enough to get you the same information, and you know, a little less flashy, but still hopefully good content. Okay, so we'll leave it there, folks. And we'll catch you next time.